Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, I have a few announcements to make. We are launching our GPCR consulting services. More information will be available soon. In the meantime, if you need support with your R&D project, understanding the pharmacology of your receptor, or need help getting an idea off the ground, email us at hello@drgpcr.com. We have fantastic talks coming up at the Dr. GPCR Virtual Cafe. Visit drgpcr.com/virtual-cafe to see our upcoming live events. Get your tickets today on Eventbrite. Don't forget, space is limited. To become a speaker or to sponsor an event, you can fill out the form on the main page of the Virtual Cafe. Go to drgpcr.com/virtual-cafe today. Are you subscribed to our monthly newsletter? Stay on top of all your GPCR news by subscribing today at drgpcr.com/newsletter. Also, to watch previous episodes of the Dr. GPCR Virtual Cafe or the podcast episodes, subscribe to our YouTube channel today. Last but not least, stay tuned for more information about the upcoming Dr. GPCR Summit held between September 13th and 19th, 2021. Visit drgpcr.com/summit for more information and how to submit your talk. And now, let's dive in. Hello, everyone. This is Yamina from Dr. GPCR, and welcome to yet another episode of the Dr. GPCR podcast. Today, I have the pleasure of speaking to Dr. Brian Bender, who is a postdoctoral fellow at UCSF. Hi, Brian.
Hi, Yamina.
It's so great to have you. How are you doing today?
I'm good. And I just want to say I'm truly humbled that you picked me to be part of this podcast. You've interviewed some true titans in the field.
I'm super excited to have you. I think the goal of the podcast is to put out there all the fabulous people who work on GPCRs, whether they've been in the field for 40 years, 50 years, or just a couple of, tens of years. And I'm glad to have you on board. So I see that you're postdoc at UCSF, and I also saw that you did get your PhD in pharmacology at Vanderbilt University. And you're not the what I would call my regular type of guest who does, cell-based assays and looks for functional selectivity in some kind of a GPCR. What do you do? What is your research about?
Well, I'm not entirely as distinct as you might think that I am. I did start in experimental work doing X-ray crystallography of all things, structural biology, not of GPCRs. When I started my bachelor's, it was the year that the beta-2 adrenergic receptor structure was determined. So I joined into science right at the cusp of structural biology revolution for GPCRs. And at the time that I was doing all those experiments, my professor, a mentor at the time, Roger Rowlett, he said those people doing GPCRs must be crazy. We were working with lysozyme, I'm sorry. Much simpler. You can just put it on the bench and it'll crystallize. But over the years, I've transitioned from experimental work. I continued in structural biology with X-ray crystallography and proton NMR and electron paramagnetic resonance spectroscopy, but eventually moved into computation. And it was really as a means to rapidly assemble all of this structural data that we had, and often low resolution structural data. Mm-hmm. And our goal was, how can we generate more accurate structures and structural ensembles of GPCRs in their different conformational states using limited structural data. And so I worked on incorporating all these different types of experimental restraints into my models. And eventually I wanted to, I was working on one or two receptors, but I thought GPCRs as a family is just too exciting. Why would you ever stop with just one or two structures or receptors, I should say? And so I expanded my ideas and my methodology to go after as many GPCRs as possible, really. And now what I'm doing is I'm taking these structures, these models that I'm predicting, and I'm asking the questions: how much can we gain from these models in terms of the pharmacology? Yeah. Can we use our models to rapidly identify tool compounds for as many different GPCRs as possible, and in particular, the receptors for which we have no tool compounds. So they are either considered orphan receptors, true orphans, in which they don't have any known endogenous agonist, but also orphan in the umbrella sense of understudied, in which maybe we do know the endogenous agonist, but it's not something that's useful in terms of asking experimental questions in a cell setting. You can't just easily take that endogenous agonist. It might be a huge protein or something like that, but it's not easily dosed into cells. So can we get new chemical probes for those receptors as well?
It's interesting that you said that you started at the bench doing crystallography. I think, and maybe I'm totally speculating, and please, anyone who's listening to this podcast at the moment, shoot me an email at hello@drgpcr.com to tell me how wrong I am. But crystallography is not, it's not mainstream. It's very specific. To me, it seems like magic. As much as, anything that's structural biology, or anything that's, as I said, not cell-based assay. That's kind of my that's where I lean towards. That's what I studied. So getting a crystal of any protein, let alone a GPCR, it's like a magical thing that happens in my mind. But I want to take you back a little bit. When did you first get interested in science?
Oh, in science. Oh, wow. So ever since I was really a young kid. As far back as I can remember, honestly, and one of the, I think, distinct moments of I think my parents realized how crazy I was about it was I joined, back in elementary school, they would have the book fair come through, to sell books to kids. And I always ran to the science section, and I joined the Magic School Bus Book of the Month Club. And every month they'd send me an experiment in a new book, and I would, dive into that. And I had a lab notebook from the age of 8, I would say, with all of my experiments that I did. And of course, we had other science experiment books. And I don't know, it was something that the concept of discovering something new was just so exciting. And it's my job now. I get to do this all the time, asking these crazy questions that maybe it's too crazy to ask, but I'm going to do it anyways, and I'm going to see where the road takes me.
And then, is there anyone in the field? Do you have any scientists in the family, or you're kind of the outlier when it comes to science?
No, my family, they have. Like my mom, she was a math teacher. Mm-hmm. So there was a little bit of the math side of things. And I took to that early as well, probably from her influence. But once I got into college and I tried out a few different majors, and I considered going into math as a major. But the first moment I got into a research lab, which was the first summer after my freshman year, mm-hmm. I was hooked, and I never left.
Was that the lab where you worked on lysozymes, or
no? That actually, it was an organic synthesis lab where we had designed that summer. We had designed, I think it was a 10-step reaction to generate some product, and in the course of the summer, I think I got through 5 of the steps in terms of all of the synthesis and purification, and then verification with NMR or whatever that we actually did it. And it was too slow. It was exciting to be in the lab, but there's so many hours you're sitting around waiting. And the fact that we only got through 5 of the steps and we had planned out 10, I needed something different. And the science department where I went to a liberal arts college. Mm-hmm. It was small, so there weren't a ton of options, but eventually I took a biochemistry class, and the professor there showed us a structure of a protein. And I just thought that was the coolest thing ever. And it explains so much of the biology, especially with regards to receptors that we had talked about. And of course, also, how chemical matter is used by cells. Looking at the structures, it all connected in one for me. And I pleaded with him to join his lab, and it took a few weeks, but eventually I got in there, and here I am.
But you persisted. That's amazing. So then when was your first interaction with GPCRs, and how come you're working on GPCRs now? Although you've been interested in math and chemistry and biochemistry and how, structures influence the function, but where does the GPCR component come in?
Yeah, well, I had heard about them having or being in the biochemistry lab and doing structural biology. We were talking about big papers that were coming out in the field. And then I graduated, the year I graduated with the bachelor's was the year that we got the full G protein coupled structure.
Mm-hmm.
And so, it was a hot paper that we were talking about just in a structural biology lab. And I took some time off or out of academia. I went and I teched in a lab for a few years to figure out if I really wanted to stay in research, if this is something I could do full term. And I joined a virology lab. Just something completely different. I had taken a class, an elective class, and I thought it was interesting. So I joined a virology lab, and I quickly learned I didn't want to do virology, but I did like doing research. And so when I was doing my rotations in grad school, because eventually I did go to grad school, the lab that I ended up joining had this GPCR project. And it was more like, I had heard of them, and they were this hot topic in structural biology. And I thought, they're giving me this GPCR project. Like, are they crazy? Do they know the gold mine that they just gave me? I was confused that none of my colleagues were fighting for this project over me. But I'd grown to be fond of them through structural biology, but have grown even fonder of them the more that I learned about their signaling and their influence in the cell environment.
So what did you work on when you got this project, and which receptor was it about?
So that original project was the neuropeptide Y receptor type 2. Mm-hmm. And in that project, what I was trying to do, and it didn't work, was or at least it didn't work in my hands. It may go on to be fruitful one day, but in my hands, it didn't work. But I was trying to label it for deer spectroscopy, double electron resonance spectroscopy, as a means to generate some distance constraints, low resolution distance constraints that we could then use to, in particularly wanted to model the conformational ensemble of states that the receptor goes through in its signaling cycle. And I had problems with the expression and purification, and we were doing all sorts of crazy things. And what was probably the real the best thing for me is that I joined a lab being open to learning any and all new techniques that I could, and I had started on a small computational project while doing this larger experimental one. And basically, what happened is, as the experimental project was becoming less and less likely, I became much more proficient at the computation, and my projects just sort of moved towards that direction. And I got quite into because of my experimental background, I was able to absorb information that was being generated by our collaborators much more quickly and translate that into restraints in my modeling, or at least understanding the theory of those experiments to know what the limitations were, so I didn't overinterpret in my models. And because of that, I glommed on to any collaboration project I could get into and said, can I please model your receptor and your ligand, whatever you're using, with the experimental data? And I, from there, just moved straight into modeling.
I love the fact that, you spent some time at the bench. So especially even within the GPCR fields as well. So it gave you an opportunity to understand how fun and yet how difficult and disappointing these receptors can be. But on a more serious note, to understand how they function and how does that data look like? And then gaining that computational experience, it's very nicely complementing one another. The bench experience with the computational, because as you mentioned, I feel like whenever you're talking to someone who only does computational work but has no background in the lab space of it, it's hard sometimes to interpret the experimental data and how do you model that.
Yeah. And then the other thing that was nice about it is, there were people that still are much better at the computer side of things than I am, even with the years of experience that I have. So I can more easily go to the hardcore coders and ask them, say, I need a script that does specifically this using their language as a way of analyzing data, and in a way that maybe my biological collaborators wouldn't be able to explain how the code needs to work exactly. They'd be using a different set of language. And so I've enjoyed being able to switch between both realms.
Which is very important. My next question was going to be, how many languages do you speak? So other than English, so you speak computational, you speak signaling, and you speak English as well. Anything else to add to the list?
Yes, in fact, French, German, and Russian to varying degrees.
Wow, that's amazing.
Yeah.
I didn't know you spoke French, so we're going to have to practice because I also speak French.
That was the first language I learned, and also the first one I forgot because it was the last one I had used. So it will be rusty, but we can do it. Sure.
Yeah. Next time we have to record a podcast episode in French, which I think is going to very much limit the audience to either the French folks in Europe or the French Canadian audience in Canada.
I love it.
But then, so you speak French, Russian, English, and some German, and, computational and signaling, which is very interesting and very important as well. And I think not everyone has the capacity to kind of juggle between, computational language and understand how you can use that data in both ways. And what are the correct conclusions, or at least the best conclusions that you can get out of both worlds. I often said this during multiple podcast episodes. I think being able to work with multidisciplinary collaborators gives you a huge advantage, because you can only generate so much data, but working together and having all these people from different fields collaborate is just phenomenal. So the work that you had mentioned on the P2Y receptor was during your grad school?
Yes, that was in grad school, and I stuck with the NPY2 receptor. Eventually moved into NPY1, and then I jumped into the ghrelin receptor, which is also super cool because the agonist is a lipidated peptide.
Yep, that's a class B.
No, it's class A. Ghrelin is class A.
Oh, ghrelin. Sorry, the
GnHR.
No, I'm with the GLP-1. But yes, that's a class A.
Oh yeah.
That is awesome. Yeah. And then what led you to the ghrelin receptor?
It was mostly the team that I was part of. So for my PhD, I was fortunate that my professor had set up this collaboration, not only with our lab, but with many labs at Vanderbilt University, within many labs at Leipzig University in Germany. And the program regularly sent scientists back and forth, primarily grad students and postdocs, to learn experimental techniques that they could then bring back into their home labs. And I had gone over to Germany a few times, mostly to work on the Y2 receptor expression and purification system for that. But I met the grad student at the time in Leipzig who was working on ghrelin, and we got to talking, and eventually it sort of turned into, he was collecting this NMR data, and we thought it was very analogous to what I was doing with the Y2 receptor. And he wanted to see a model of how his agonist was binding to a receptor so that he could generate hypotheses on where to probe. And so we said, let's model this, and the bosses were into it. So we did it.
That's awesome. And you also got to practice your German.
Yes.
And although I'm pretty sure everyone speaks English in Germany.
It's actually very difficult to practice your German in Germany because everybody there wants to practice their English on you, and they are 10 times better than you at English than you are at German. Yeah.
Oh God. And then so you completed your grad school. What led you to UCSF?
Oh wow. Yeah. So actually, Brian Choiquette, the professor whose lab I work in, he came and gave a talk at Vanderbilt in one of my first couple of years, and talking about how different proteins may be connected by the type of chemical matter that is recognized in their binding pocket, and that pharmacophores are better conserved than full proteins. And so it's a nice way of looking at off-target effects. For example, if it shares a similar binding pocket in a different receptor, it's likely going to have an off-target there. And so he gave that talk early in my PhD career, and then I kind of forgot about him. And over the years, because it wasn't a GPCR talk, right? And over the years, I was going to GPCR conferences, and I was seeing what he was doing with discovering chemical matter for these receptors. And it was the exact application that as I'm writing up my thesis, thinking, what are the next steps for the methods that I've set up? What can they be applied on? I thought, this is it. I want to use my models to find compounds for as many different GPCRs as possible, and the fastest way of doing that is going to be an in silico approach, and in this case, virtual docking. So I asked, and he said yes, and here we are.
There you go. Yeah, and you were talking about going to conferences, and I think this is a time to plug in the conference, the Gordon. Why? I think it's the coffee this afternoon. The Gordon Research Conference. Can you tell us a little bit more about what you're organizing and when it's going to happen?
Right. So I am one of the co-chairs for the Gordon Research Seminar in Molecular Pharmacology, which is the pre-conference to the Gordon Research Conference. This is the seminar series is intended for the early career researchers, so primarily undergraduates, graduate students, and postdocs.
Yeah.
If there are anybody that's more senior than that, there's a good chance they're there for a career or mentoring workshop. But the idea with the seminars is to give the speaking time and the platform to the people that are doing the science. Yeah. The ones that are spending the hours and the weeks and the months at the bench trying to get their protein to express so that they can then do whatever fun experiment they want to, and figuring out all the reasons why their receptor doesn't express when the next one over expresses so easily, right? What are those subtle differences? So this is really giving them a chance to be the ones leading the scientific discussions. But also, one of the things that I've experienced myself as I've gone to more of these early career researcher conferences, that these are my future colleagues. At some point, the old guard is going to retire. Yep. And we're grateful to them for making the discoveries that they did. But the next generation is ready to take on the challenge of the big open questions in this field. And so I go to these conferences, and I'm thinking, I'm going to be seeing you for the next 20 years. And I'm really happy that you are part of my community, and that we're going to make these connections, and maybe we'll be future collaborators and write a paper together or a grant together or something like that. But I'm there, and I'm talking to all these people who are excited about GPCRs in their own unique way, whether it's signaling or expression in different diseases or under expression in different diseases. Yep. And then, I'm coming from the structural point of view, the computational point of view. And it's just this big, lovely mixing pot of ideas. And, I always walk away with a conference with at least 10 new things I want to try in my own work. And so, we're going to have this conference. It's scheduled for the summer in Switzerland, and we're keeping our eyes very closely on the COVID situation, as you can imagine. The Gordon Research Conference board recently did determine that, or they at least project that our conference will be able to go forward. They think that the caseload will be down, and people will be able to travel, hopefully also willing to travel. We have to see, but I mean, at this point, we have to plan the conference as though it is going to happen. And so we're actively fundraising. We're accepting applicants to that want to give posters or give talks, and all of that. And of course, if it is determined that we can't go forward with this conference, then basically we'll just move into the next year's planning. There won't be an end to the Gordon conferences anytime soon, just because of how important they are to the field. It's one of the few places that you can go to such a small conference and share unpublished data and get feedback from the people that are going to be the most helpful.
Agreed. I've been to a couple of Gordon conferences and also research seminars, and I think both are so one, fun to go to because of the diversity of the crowd, but also it's a small conference. I don't think there is more than 200 people. So you have to apply and register as soon as you can, because you need to be approved to go to the conference. But I think the diversity in research and topics, especially with the trainee seminars, is just phenomenal. And I've gone home from a Gordon research conference, and it ended up in being a collaboration because someone saw my poster who said, "Oh, wait a minute, my roommate could use your expertise," and then we ended up collaborating. So I think it's a phenomenal thing. Plus, usually these Gordon research conferences are held in super nice places. Where will this one be?
This is I'm gonna butcher the name. It's Les Diablerets in Switzerland. It's a mountain resort in the Alps. There's a river that runs through a valley that you can walk by in the afternoons, or you can go hiking. I'm guessing since it's the summer, there won't be any skiing.
Still sounds like a magical place filled with, little elves and a lot of GPCR work.
Exactly. All the GPCR fairies will be out sprinkling their wisdom.
Well, hopefully with now. So for those of you listening, we're recording this episode at the end of December of 2020. And just last week, the Moderna COVID vaccine was approved. So the Pfizer BioNTech vaccine has been approved and has been started to distribute. And today, the European Union also approved the Pfizer BioNTech vaccine. So hopefully, next year is going to rain vaccines everywhere, and then we can finally go back to a somewhat normal way of doing these things.
We sure hope so.
I really hope so, that we can just, travel again, be together again. Although I have to admit, having virtual conferences this year was really a blessing. Thank God for the internet. Thank God for platforms like Zoom, because we were able to still talk science in spite of the fact that we were just stuck at home.
No, and it was fantastic as well for, in particular, the early career researchers, because it was free. Then to go to these conferences, and we're always struggling to find the travel grants or whatever to get sponsored to go to these conferences. So that can always be a limitation. But also the conferences that I was able to attend, some of them I'd never heard of because they were European conferences or Australian conferences, and it introduced me to a community of researchers in the field that I would have had few interactions with if I had to, travel as far to hear them for 2 days.
I agree, especially you can't travel. I mean, it would be a miracle if as a postdoc or a PhD student, you can go to Australia for a 2-day meeting from the US or from Europe. I mean, unless you win the lottery, it's kind of hard to justify to your boss or even to a committee to spend so much money and time to just travel to those conferences. And I think it was a great year in that regard, where, as you mentioned, you get to know different groups from all over the world who have a similar interest to you. So what I wanted to kind of highlight is that you're co-chairing this the Gordon seminar, research seminars. I'm having trouble. I for some reason I want to say GSR, but it's GRS. Gordon Research Seminar. And that takes us to the next topic I have. So you're a postdoc, you've been working in the lab, you've done computational work. What led you to take on what would call kind of extracurricular activities? And what did you learn by co-chairing such a conference?
Oh, wow. I've always been involved, I guess, in extracurricular activities, if that's how you want to say it. I mean, what I love about what I'm doing here for this conference and for any of the other programs that I've been involved with in grad school or undergrad is it's really about connecting scientists. I mean, I love science. I love doing science, and I love hearing about science and reading science. But it's really about bringing scientists together. And so I serve on the Postdoctoral Scholars Association at UCSF, and it's about how do we create a community of scientists, because the more people that are connected and plugged in, the more people feel that they are a part, that they belong to that group, and then the more likely ideas are going to flow. And so that's really, why I even ran for this position is, how can I facilitate these conversations and these connections? What have I learned? Fundraising is interesting in a pandemic. I mean, fundraising is interesting as it is. I'd never had to do it for a conference. And I don't think I'd fundraised since I was a Boy Scout and selling popcorn door to door. So writing to companies for thousands of dollars was very intimidating. But during a pandemic, it's even more so, just because there's so much uncertainty. And I have to wear multiple hats of I am myself this anxious person who's not sure if I'm going to be able to travel, but at the same time, I have to treat this like it's actually going to happen, and put in the legwork. But I've been working with just some fantastic scientists. So Dr. Nikki Perry, she's the co-chair with me at the GRS, but then also Andrew Tobin and Martine Smit, and they're both from Europe. I've been connecting with them every other month in meetings, which they may have remembered me from some conference prior and said hello as, oh, we recognize you, but not actually know who you are. So I'm able to make some very nice connections with people who I may have otherwise not been able to.
Yeah, I think the term extracurricular, I use it just because of a lack of a better term. I think involving yourself as a scientist, whether you're a junior or you're an established scientist, in organizing meetings, in meeting people, is something that is as important as being at the computer or at the bench, because you cannot do science on your own.
No.
You need to collaborate. You need to talk to people. Sometimes, you, as you mentioned, you were having trouble labeling the P2Y receptor. Well, maybe it wasn't a good match between you and the receptor, but being able to go to Germany and get to work on the ghrelin receptor and also have a side project, which was computational, really kept you in the field. The thought I have is, think about it. If the only thing you had to do is stay at your bench and try and label this receptor, I don't think we would be here today.
And I think that highlights a lot of the struggles that people are going through in this year in particular. I haven't seen one of my colleagues since the first week of March in person. I haven't seen the inside of my lab since the first week of March. I haven't been allowed back into the building. It's one of those blessings and curses of the computational sciences is that we were the most equipped to transition to working from home. But at the same time, because we have this relative ease, if the cluster goes down, you can't do anything. But since we do have this relative ease of being able to work from home, we have been asked to just completely stay home. And at the moment, that for me extends until the summer of next year.
Wow. I guess it can get very lonely.
Yeah,
but I invite...
For sure. But I mean, that's why these virtual conferences have been great. And, the fact that I even have this extra work of organizing the GRS, that's something that keeps me still connected. The podcast that you put on has been, I haven't been able to have these in-depth conversations with other scientists because I haven't been late night at a conference
Yeah.
in a year and a half.
Yeah.
That doesn't translate as easily to these virtual conferences as I'd hope to. So, I listen to your podcast, and it's me living vicariously through you, listening in on these conversations. So we have found these just truly incredible ways of adapting to the situation around us. I'm so looking forward to going back into the lab. I can tell, even if it's just to sit at a dusty desk, to clean my desk for the first 2 hours or something. That's going to be so exciting to know that I'm soon going to be surrounded by my colleagues.
Yeah, I feel like as a community or as humans, I feel like we're kind of, and please excuse the nerdiness in this example, but you were talking about, the fact that we adapt. And the only image I had in mind is, a G protein knockout hexa that adapts without that G protein and finds other ways to signal. And I think that's what we did as a community and as a world this year.
I think you're allowed to be nerdy.
Thank you.
Go for it.
That's the thought I had when you were talking. And I think it's a great way to cope with the fact that we're all at home. And, even those who go to the lab, I have colleagues from NIH who have, 2 days out of 5 in the lab, and experiments take 5 days. So they really rely on each other to start rolling the experiments. But then again, it limits you. So there is no space for error. You have to go in, you have 4 hours, and that's it. So it's a hard thing. I'm glad that the field has still been very busy. A lot of publications coming out. And I'm confident that us going back to the community, to the lab, to conferences, it's just going to make the field boom even more.
I think one thing that may have come out of this virtual setting and more different people showing up at these conferences is that we'll have new collaborations formed that wouldn't have otherwise formed if we had stuck to the traditional route of you do science in your area and then you go to a conference and you meet somebody there. The people that couldn't go to that conference previously will no longer be hindered the way they might have.
Exactly. So my next question to you, since we're talking about the future and going back, what's in the cards for you once you finish up your postdoc at UCSF?
Oh, so many things. This is always the worst question, and you get it at the end of grad school too. What do you want to do when you grow up? And that's always so difficult to answer. I've always been drawn to academia with the idea of paying it back to the next generation. I enjoy teaching. I enjoy basically translating concepts from one language that is maybe too complex to another language that is less complex for young students to then get excited about the fields themselves. And so, professor position is definitely something that I would enjoy. Mm-hmm. That being said, as the year has gone on, there may be changes in the future of academia. Who knows what the future holds? And if this year has taught me anything, it's to adapt. And I've always been open, if not tepid, with the idea of industry. But the more that I've actually interacted with industry, through collaborations and through industry partners that I've also met at conferences, it's like, wow, they really do amazing work there and really cool projects, and to be in that type of environment where everybody's supercharged and excited, I think is also equally appealing. And so one of these days I will find out what I want to do when I grow up. But I just have to wait for that day to happen. I love doing science, and whatever allows me to keep doing science, if that's in academia, if that's in industry, if that's in a nonprofit, I don't know. Like, let me see where that leads to.
I think exploring is a very important thing. And up until the pandemic hit, if you would have asked me the same questions that I just asked you, I would have said, well, I don't know, or I'm not sure. I was very happy for a very long time at the bench. And then I got to a point where I felt like I could be doing so much more, or at least get, I would have loved to get more data faster and advance the field faster than only rely on my own two hands.
Yeah.
And I don't know if you're like me, but when I'm passionate about a question, I mean, I'm doing experiment 1, but that brain is already at experiment 10.
Oh, exactly.
And then it just all gets all messy in there, and I have to write it down because that's what I'm thinking. And I got kind of frustrated. So with the pandemic, although it was an odd year, not only for me, I'm sure for everyone, coming up with the idea of the podcast was a revealing moment for me. And it's so interesting. I don't think I said this to anyone, but I was trying early February or January to figure out a way to do something that has to do with GPCRs, but it's very far away from the lab. And on a Saturday morning, while waiting for my little one to come out of his jiu-jitsu class, sitting at a Starbucks within a Target in the Boston area, I was reading an article and I thought, a podcast, that'd be really an interesting thing to do. And there were no podcasts on GPCRs. And there you go. A couple of months later, I'm humbled. This is, I think, episode 35 or 36 that I'm recording.
Oh, wow. That's amazing.
So I logged in almost 400 hours of podcasting, recording in a year, and I could not be happier.
Yeah. You sound like you have an incredible job. Like I said, you get to talk to all these people, and I'm living vicariously through you, through your podcast. And the fact that, it was something that you maybe weren't thinking of, and the idea came to you in some random moment, and you ran with it. I think that is truly the mark of anybody that is a great thinker. That's going to sound terrible when I say I also like me, and then people are going to think I just wanted to call myself a great thinker. But I would have so many ideas when I'd be riding my bike to and from work, because it's, you can't do a whole lot when you're riding a bike, because you have to focus on the other cars that might be running you over. So you can't listen to music, you can't talk on the phone, like you are in the moment. Yeah. Riding a bike. And so your mind just starts running away with, what's the experiment that I just did, and what was the data, and you start processing it and thinking of the next idea. And I got into such a habit of writing stuff down. Kind of like you said, I'd have so many ideas, I had to start writing stuff down. And probably 9 out of 10 of those ideas were stuff that were just silly, or we definitely didn't have the technology to do. But if you write enough ideas down, there's bound to be some good ones. And I would say half of my thesis, maybe a third of my thesis, came from an idea I had while riding my bicycle, that I decided to go into the lab and tinker with. And it turned out to be this whole cool method of predicting structures. So I say go for it. Go with these wild tangents.
I think it's something that could be a really good way for younger scientists or junior scientists to, think a little bit outside of what you're doing on a day-to-day basis in the lab, and take some time to think. And as you mentioned, just write down ideas. And if some idea sticks in your brain, and if weeks later you're still thinking about it, even though everybody tells you no, it's not a good idea, why not try it out?
Yeah. Actually, I got the idea came from this former chair of the graduate department at Vanderbilt, Lee Limbird. She wrote a very nice book on receptor theory that I have read many times. I was unfortunate enough that I didn't actually get to work with her while she was still at Vanderbilt, but she came back for one of our department retreats to give the keynote talk. And, she was this idol at this point because I'd read her book so many times, taking in all of this receptor theory. And I was like, oh my gosh, we get to hear her talk. And she told us that every month when she was a professor, she would put on her office calendar that she was working from home. Mm-hmm. And she would tell her family that same day that she was going to the office. And what she did is she went to this lake in Nashville, Radnor Lake, which is this beautiful, outdoor, picturesque scene. And she'd spend the whole day with a notebook in hand and just walking around and sitting and thinking. And she reflecting on what she had done, what the next thing she wanted for the lab to do. And so I kind of took that philosophy forward with, my bike rides or any of the miles of walking that I've put in 2020 as ways to, keep myself sane. But yeah, it's she taught me, without knowing that she did, that, taking time to think about what you're doing is way more valuable than just trying to do the next 10 million experiments all at once.
Agreed. And I think if I remember correctly, she was the first hire of Bob Lefkowitz at Duke.
That's beyond my knowledge, but
I think
I'd be willing to believe it.
I think she was, and I've heard stories, which I think by the time this episode airs, everyone will have heard that we're starting out season 2 of the podcast next year with Bob Lefkowitz.
Oh, wow.
And yeah, you'll see how phenomenal that episode is. But I think she was the first hire when Bob got to Duke. I'm not even sure he was a PI then. I think he was a postdoc, almost becoming a PI. But what a small world that, she's the first hire in Bob's lab. And then, she has this phenomenal career. And then you read her book and then you learn from her, which I think it's kind of, going down generation by generation.
Like I said, we stand on the giants before us.
Agreed. We're kind of all learning from each other. And I think that's the whole point of it. Yeah. What you had mentioned when I asked you the impossible question, so what do you want to do when you grow up? And I've talked about this on multiple podcast episodes, and I think exploring your options is never a bad thing. And I like the fact that you have, you have multiple avenues, and being able to, co-chair conferences, go to conferences, go look for funding in a pandemic year. Still, it's a challenge, but you're going to learn a lot from it, or you have already learned a lot from it. And getting those connections is just crucial for your career, because you never know who you can help out, and you never know who can help you out in the future.
Exactly.
That's phenomenal. And so if you were to go into industry, what type of positions? You said you love science and you really want to continue working with a great team where people are psyched about the project they're working on. You would want to stay in the computational / GPCR realm, or just keep on with the computational work?
Yeah. It's hard to ever leave the GPCR world once you go into it, just because the longer you stay in it, the more you realize we still don't know in this family. There are a lot of receptors in this family. A number of orphan receptors in the non-odorant family, and then of course the odorant family being just a huge number of receptors that we still haven't explored. And so it would be tough for me to leave GPCRs at this point. I just feel like we're at such an inflection point in terms of structural knowledge, of being able to understand how drugs really work at their targets and in their signaling pathways and all the various different signaling pathways, and whether you're even studying the right pathway because you're doing in vitro versus in vivo, and everything that comes with that. I mean, we're all asking these questions that is just going to lead to really cool new knowledge. So I don't think I could leave GPCRs. But I think in terms of the types of science, I think computation is going to serve a huge role in getting us to this next level of knowledge. If we keep asking these questions at individual receptors and developing techniques that work at individual receptors, while we keep gaining knowledge doing that, we aren't going at speed across the family. If it takes us 3 years or so, let's even just 6 months to a year to find a new chemical probe for an orphan receptor. If you multiply that out across all of the orphan receptors, I mean, you're still looking at decades, right? And we have to think grandly when it comes to these challenges of, what can we do to de-orphanize as many as possible. And David Glorian's doing some great work there with Alexander Hauser. But even more broadly, once we do identify their endogenous agonists, they may not be useful in an in vitro setting, right? So we need the chemical probes that we can really, probe the biology of these receptors. And so I think I just think computation's the place that's going to get us there fastest.
I couldn't agree more, to be honest. I think, working at the bench and coming up with assays that relevantly track the function of a receptor or a family of receptors that you're interested in is important. It's great to have those tools, but the power of computation, the combination with computational work is just, takes it to an exponentially next level. That absolutely is crucial, especially now that, we have all these methods, these experimental methods that allow us to acquire data so much faster. And not only endpoint data, you can acquire kinetic data. You have robots that, just dispense a bunch of molecules in there, and then you can measure multiple signaling pathways at the same time. So the human brain at some point cannot process all this information. And you need a computational.
It gets saturated, but I mean, you do raise the valid point that, I mean, what the computer is doing is taking real data that somebody has generated, and synthesizing it and creating new hypotheses. I mean, we are still completely in debt to the people doing the bench science.
Oh, absolutely. I think we're working hand in hand.
Exactly.
With everyone. Anyone who's working at the bench is as valuable as someone doing computational work, because the computational scientists cannot do anything without well-controlled, well thought out experiments, valid data, and then you can generate as much data as you want. But if you don't know what to do with it, it loses its value. Just wasted time and energy.
Yeah. And I mean, going back to being able to speak both languages, sometimes I have to switch back into the biologist language when I'm trying to convince my collaborators, like, I need this type of experiment so that it answers this question much faster than, maybe another, whatever they had on the shelf. And then again, like I said, when the data does come back to be knowing the theory, the how that experiment worked, if I can use the EC50s to mean a certain thing, or if I can use the Bmax to mean a certain thing, whatever it is, make sure that I don't over restrain the models, come to false conclusions because I didn't treat the data that was generated properly.
Absolutely. So before I let you go, I have a couple of more questions for you. Question number one is, so we've talked a lot about the work that you do and how much you love science. And we also talked about the fact that you need to take some time off from the bench or some time off from the computer to come up with ideas and let your brain actually do what it needs to be doing to advance, your work. How do you manage your everyday work-life balance? I don't like this term, but that's the best term I can come up with. The reason I don't like this term is because you're the same person who is outside the lab and within the lab. So it's kind of how do you manage your non-work activities in parallel with your work activities?
Yeah, I mean, that's obviously gotten much harder with the pandemic. I mean, pre-pandemic, I was a volunteer at a science museum. I was in a band. I would go hiking. At least I can still do hiking. But I mean, I had these other things that I was involved in as ways of giving my brain that complete turn off and complete other focus from the science. And so, now when I do step away from the computer and I go on my walk, I often still think about the science, which is not a bad thing. But eventually your brain will get fried. So, the only things that have really kept my work-life balance sane is I make these schedules for myself. Mm-hmm. Like I will wake up at a certain time. I will start working at a certain time. I will, take a walk at a minimum once a day. Something that gives me a semblance of this. There is a work life and there is a not work life, even though it's the same, four walls around me at any point. It's more like I had to create that structure in order to achieve that balance, because early on, I just found myself from morning to night looking at a screen, whether it was for work or if it was to watch a movie or something. And I realized this can't go on for 9 months, a year, however long actually, it is going to be. So in particular, scheduling those breaks has helped.
I think it makes a lot of sense to be able to, have those breaks. And sometimes it's difficult. And I know I'm the type of person that whenever I'm in front of a computer and when I'm analyzing the data, I can't stop because I'm too curious to see.
Oh my gosh. And that's actually the worst, best thing about working from home is there have been so many nights I've caught myself, it's 2 a.m, because I went on like a reading binge, or I was analyzing data a certain way and I found this new idea and I wanted to, explore it. And so it's great that I'm able to play in that sense, uninterrupted. I don't have to run to the bus to catch my ride home or anything like that. But on the flip side, if I do that more than once a week, or my body says, nope, that you can't, you're not that young anymore. You got to go to bed.
Exactly. You're not 20 anymore.
Exactly.
Where you could go, full week with sleeping 4 hours each night because you had exams, and then you could sleep a week to recoup that time. But I can definitely relate to the fact that, once you're in front of the computer and then you're trying to figure out, okay, what does the data tell me and what are the conclusions, and let's try it this way or let's try it that way. And then you look up and 4 hours just went by. And you've been thirsty for the 4 hours, but you've been telling yourself, oh, let's just this one more. Or the times
that always gets you, that one more. I'm just going to do this one tweak. Yeah,
exactly. Or I think worse, as a person who's worked in the lab for such a long time, it did happen to me that in the middle of the night, I woke up and I was dreaming about redoing, running the 50 plates that I ran the day before. And then remembering that at plate 48, I made a mistake at 12:38. And I was like, oh God, we can correct this, but just go back to sleep. It's not conductive at this point. It's done.
Yeah, I think we have all suffered the waking up in the middle of the night, remembering something that you did and particularly wrong in some experiment.
Exactly. So the next question I have for you is any advice for junior scientists, for young scientists who want to contribute to the field and, who are kind of exploring their options, whether they should continue working at the bench or go computational? Any take-home messages for them?
Yeah, and it's one that I have to remember myself because sometimes I'm just not as good as I want to be. But it's, when you do have the opportunity to go to a conference, or even if you don't, if scientist is coming to give a seminar at your school, ask to have lunch with them. Even if you don't know their science, ask to have lunch with them, because usually about 5 minutes into talking with another scientist, even if they are in a completely different field, you find some similarity. Like, you all forgot to make a PBS buffer correctly one time and it ruined your experiment. It's something simple like that. And then you have that connection, and then you're off to the races. And the more people that you feel comfortable talking to in the field, the more you feel just connected to the field, and that you can start coming up with some ideas that you think, wow, this would actually really contribute to their knowledge, because I know they're actually struggling on this, and I have this new way of thinking about it.
I like that. And I think that takes me to the kind of to the next point, is that when you talk to other scientists, you realize that you're not the only one who had made a mistake making the PBS based buffer, or you ran your DNA samples in water instead of the TAE, and you were wondering why the bends were diffusing in the in the gel. So I think realizing that we're more alike than different is also something that keeps you sane in the long run.
And in particular, if you have those sort of conversations with the people that you maybe idolize, or you think they are the gods in the field, and then you realize they're just as human as you are and have made the same types of mistakes and are just as human, it makes you just appreciate the field that much more, appreciate the community that much more, and feel, in particular, confidence to speak your voice.
Exactly.
That's what I think a lot of young trainees struggle with is confidence to ask questions, to put out forward opinions.
I think one of the goals of the podcast as well, especially lately with the last couple of episodes, has become to show how human we all are, whether you've been in the field 5 years, 20 years, 25 years. We are all human. We all struggle. We all have to sleep. We all have to work hard. And I think the best way to kind of evaluate and compare yourself is to compare yourself to yourself from yesterday or the week before, and not, tell yourself, oh, I can only do this type of assay, but, the guy next door or the PI 3 doors down is so much better than I am. You don't know what kind of struggles they went through to get where they are. And I think you should just focus on improving yourself through hard work, yes, but through smart work and through talking to people and realizing that we're just human. We're just human.
I in particular, you say I in particular miss these social hours from my graduate school where it was designed for anybody that did structural biology. Mm-hmm. Didn't matter what type of structural biology, but if you did structural biology, come to it. And I can't tell you the number of experiments that I had troubleshooted by another scientist who had the same problem. And then I go back into the lab the next day and I try whatever trick they had, and it was done. So everybody has struggled with some assay at some point and has the trick that they used.
Exactly. Whether, it's standing on your left foot and jumping around to make your experiment work, or talking to a colleague who says, well, you should just, add some BSA or add some sodium, 5 millimolar, and then poof, it just
it's amazing, simple things like that are that make all the difference.
Agreed. So last question, which I ask to everyone, any aha or Eureka moments that you had during your trajectory as a scientist?
Yeah. I very briefly mentioned it earlier. When I started the PhD project, I was working on a single receptor, the NPY2. And then because it is itself part of a family, there's 4 of them. I was, thinking, what do I know about NPY2 that I could translate to NPY1?
Yeah.
And then when I picked up the ghrelin project, I was like, well, what do I know about these 2 receptors that I can translate into ghrelin? And that's when I started thinking broadly. It's like, if there are things that we can translate between different receptors, how much can that be applied to the whole field? And I started doing these multiple sequence alignments for, larger and larger and larger families. And I, having come from a experimental background, and I didn't know how to write very good scripts at the beginning. So I had to analyze a lot of the data by eye and by hand, because I didn't know how to write a for loop. I was very naive when I started in computation. And so I would stare at these multiple sequence alignment tables on my computer for, days, and I would relate them to the known structures, and I'd map out the carbon atoms of the sequence alignments, and I really started seeing these connections between families through their sequence and their structures. It was 6 months of probably just being silly, not taking the time to write the code, but looking at the data as closely as I did, it honestly, it was beautiful. And I was like, I have to go into more receptors. I can't stay with just the one.
But so I feel like at some point you were only seeing a bunch of letters coming and going on the screen and trying to figure out what it was. It's kind of like reading a sequencing gel, which not sure a lot of people know how to do that. And I remember struggling with it, but you don't want to do that anymore.
But if you don't know how your data is being used.
Yeah, that's true. That's true.
Yeah.
But I think it still gave you.
I'm glad I know what I know now.
There you go.
And write the scripts that I can.
There you go. Well, all right. Thank you, Brian, for your time. I really enjoyed our discussion. And if I don't speak to you until next year, well, happy holidays. And Happy New Year.
Happy holidays to you. And thank you so much for letting me be a part of this.
Of course. And for those of you listening in 2021, well, hopefully by the time you listen to this, the widespread vaccine will have made its effect. And hopefully we can all see each other again soon at a conference near you. Have a good one. Bye-bye. Bye, Brian.
Bye.
Thank you for joining us for this Dr. GPCR podcast episode. I'd like to thank our guest, as well as our team members, Attila Forrest, Shivani Sajdev, and Jin Chung. Please subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcasts. Another way to support us is to share the podcast, the newsletter, and the virtual cafe with your colleagues. Email us with any questions or suggestions at hello@drgpcr.com. Until next time, stay safe.